Okay, so welcome everybody. It's a great honor for me to introduce today Sabine Flitsch and her lecture on carbohydrates and carbohydrate sequencing. So please join me with a big hand for Sabine.
Thank you. Okay, so if you remember in my first two lectures, I gave you a general introduction to biomolecules. So we talked about nucleic acids and small metabolites. And as I said in my first lecture, I'll now spend a couple of lectures on carbohydrates because that's my passion and my research interest. So the style of the lecture will be a bit different as well, in the sense that I will go through some of the fundamentals, because I know not all of you work on carbohydrates, but although I know some of you are experts. But I also will talk a little bit about our research, to bring it to a more advanced level. And hopefully there's sort of something for everyone in this lecture. So this is the first lecture on carbohydrates. Today I want to talk about sequencing of carbohydrates. What methods do we have for structural elucidation of carbohydrates? And then tomorrow I'll talk about enzymes and also arrays of carbohydrates, so more sort of biological applications. So today is very much on physical methods. How do you determine the structure of carbohydrates, and what do the carbohydrate structures actually look like? So it will be actually quite chemical in that sense. So let me just introduce you to carbohydrates. Okay, so again, if you look at the dogma of biology, you start with sort of tissue and cell lines, and then you have your nucleic acids, and then you have your proteins, and then after the proteins come all these metabolites. And nowadays, we have got such good tools for analysis that we can actually analyze these things sort of on a genome, what we call genome-wide basics. And obviously, you're very aware that there are these areas of science which we call the omics science. So these are genomics, transcriptomics, proteomics, and then, of course, after all those come the omics for all these metabolites, so glycomics. So glycomics mean we're trying to understand all the carbohydrates in terms of their function in a particular cell or organism. But also there's metabolomics, lipidomics, and so forth. So people, just labeling their areas now, which are much more data rich, so we can get a lot and lots of data as omics, as you can see. And you can see carbohydrates sit very much with these sort of metabolites. And that is, of course, because they're the class of biomolecules, although they're biopolymers, very much like the nucleic acids and the proteins, the biopolymers, long polymers of mono building blocks. They actually are not directed encoded by the DNA, whereas you can predict what the protein sequences, not function, but sequences look like in an organism if you just sequence a genome. And we've gone through that in the last two lectures. Glycomes, you can't actually determine a priori at the moment. You can't just look at the genome and say, okay, these are the carbohydrates that are in there, despite the fact these are all long polymers as well. Of course, lipids and metabolites are not polymers anyway, they have to be made. But the sugars are also made through these biosynthetic pathways, very much like, glycolysis and citric acid cycle and so on. We have biosynthetic pathways which generate them. And again, each step is generated in carbohydrate biosynthesis is generally catalyzed by a dedicated enzyme. Okay, so we need to know sort of what the enzymes look like that actually make the carbohydrates. So we don't have this direct link between RNA and carbohydrates, and that makes analysis very, very difficult. As I said, we don't really have to sequence all the proteins in an organism, because we know the sequences, because we can read it through the genetic codes straight from nucleic acids. Carbohydrates, we just can't read the sequences, even if they're long polymers. And so the carbohydrate sort of structureomics, glycomics, is very much dependent on good analytical facilities and methods. And I'll talk a little bit about sequencing of carbohydrates. What is the function of carbohydrates? Nucleic acids, we said, they encode all the information we need. This is the genetic makeup of an organism. Proteins that already introduced to you, they have a structural role, but also very importantly, they make up all the enzymes. So they're all the catalysts that catalyze all these reactions. What do carbohydrates do? Again, they have several functions, but one very interesting function. So they, for example, make up all the polysaccharides, cellulose, starch, chitin, all these big classes of polysaccharides are carbohydrates, of course. Then you have the small sugars like glucose, sucrose, fructose, and so on. And obviously these are massive amounts of material. In fact, actually, you could really claim that the biggest biomass on Earth is all carbohydrates, and that's obviously due to cellulose and chitin and all these polysaccharides. But in terms of interesting biology, the carbohydrates also have very specific, sequence-specific biological roles, and that's sort of something people who study glycomics focus more on. And this is a sort of a picture of a cell surface. You'll all be aware that all cells have something called a glycocalyx, and you can stain the glycocalyx and see it under the microscope. So this is sort of part of a cell here, and if you stain it, you see this layer, blue layer, for example, if you use blue stain, carbohydrate stain on the surface, and that's the glycocalyx, and that's generally mostly carbohydrates. It's quite a sort of unstructured a surface. And every cell people have looked at in biology has a glycocalyx. So all cells in biology have this glycocalyx. Not all cells have nuclei. Red blood cells don't have nuclei. They all have a glycocalyx. So this is obviously biologically extremely important. And because it's sort of so diffuse and doesn't seem to be very structured, for a long time people thought, well, it's just sort of a sugar coating, Of course, cells have lipids, and, lipids don't interact very well with the aqueous environment. Why do we, maybe we need sugars just to sort of protect the cell, keep it sort of together, coat it, make it act as a barrier or as an interface to the aqueous environment, which would be out here. But actually, people now found that there are very specific structures which have very specific biological function, and that's often, if I can lose this, often shown in a cartoon. This comes from my colleague Stuart Haslam at Imperial College. Generated this cartoon, but you can find it actually on the internet in many different forms and shapes, and that sort of illustrates what the carbohydrates in this glycocalyx actually do. So as I said, they sit on this cell surface, generally as conjugates, so that you don't tend to get free carbohydrates. So for example, you have a lot of glycolipids, and you've got a lot of glycoproteins, so they're linked to proteins, and I think somewhere maybe there's also a picture of a lipid, and so it's these little cartoons here that are meant to show the sequence or the specific carbohydrate that's responsible for these interactions. So we now know that cell-cell communication is extremely important, of course, and it's mediated by carbohydrate, and generally by carbohydrate-protein interactions. So if you study carbohydrates, you often have to look at proteins as well. So it's people who study carbohydrates tend to know a lot about proteins as well. So generally, we're talking here about carbohydrate-protein interactions. Some people have proposed carbohydrate interactions, but there are not many examples, if any. So that's still an area very much under exploration. Generally, you're talking about having a protein that's a glycoprotein. You've got the carbohydrate on the protein, and then it interacts with what you could call it a receptor, so carbohydrate receptors, or we call them carbohydrate binding proteins, or sometimes also lectins. So lectins are a class of proteins that bind to carbohydrates. And you can see, first of all, these are involved in cell-cell communication. So in particular in immunology, this is extremely important. And for example, in innate immunology. but also proteins, so hormones and enzymes and antibodies. Desilylated glycoproteins can mediate this, and the carbohydrate can be either on the cell surface actually, or on the cell surface you've got a carbohydrate binding protein as shown here. Lectins, these are these carbohydrate binding proteins, but also external agents. So we now know that bacteria, for example, and in particular viruses, really invade cells and tissue through carbohydrate-mediated interactions. Very famous example is the flu virus. And the flu virus, those of you who know better recognize the sialic acids. I'll show you the structure in a minute, and in a very specific form. And it needs to attach to the sort of endothelium in our airways through this carbohydrate interaction, and then can invade the cells. So these are very important. And then obviously is very important biology. The nice thing about carbohydrates, they're sort of quite unstructured, they're quite small molecule, and if you understand these interactions, you can immediately think about therapy. So you might want to interfere with cell-cell interactions. Well, you could think of generating small molecules that mimic these carbohydrates and that interfere with these interactions. And this principle is being used very successfully for antiviral. So the anti-flu virus medicine that are targeting this particular interaction, this interaction of the virus with the cell surface. So really this leads immediately to medical applications, and there's a huge amount of work at the moment on making new antibacterials. You know that especially in hospitals, antibacterials are failing now because you've got resistance of bacteria, and there's a large body of research which looks at trying to use sort of carbohydrate protein interactions as targets, for example in vaccines. So the bacteria would have carbohydrates on their surface, and then vaccines would be raised against these the surface bacteria, or you would actually use carbohydrates as protecting yourself against bacterial infection. And I think there was even is a project going on here in looking at that. So so you can see here, this is actually very, very interesting specific biology beyond carbohydrates. So carbohydrates have many roles in biology. And then, in addition, what's made carbohydrates particularly a hot topic lately is this protein here and other glycoproteins. And I'd shown you this picture before. This is a glycoprotein called EPO. This stimulates red blood cell formation. It's a natural hormone in our body, and it's a billion-dollar drug. It's a beautiful story of medicinal chemistry, where someone recognized that EPO in our body is the key hormone that regulates red blood counts. If you inject EPO into people, their red blood cell count gets up. So they produced this, a company called Nanjing in California produced this protein, and they found that actually this protein here can be expressed, and it actually even interacts with its receptor, the EPO receptor. The biology is quite, but it only really is active in the body if you put these carbohydrates on, and that's to provide stability against degradation and also targeting the protein. We understand this now, and you can see actually the red carbohydrates make actually a large part of the glycoproteins. So obviously this particular protein is not a cell surface protein. That's why I sort of added it to my introductory lecture. So actually the analysis and the synthesis of glycans for glycoproteins is incredibly important. And this is really, as I said, especially Western pharmaceutical companies really focusing on these glycoprotein pharmaceutics. I've shown you a list in my last lectures that they're the major drugs now on the market. So they're incredibly profitable for Western pharmaceutical companies. So EPO is a biopharmaceutical. 40% is due to glycosylation. It's produced recombinantly. Obviously, you don't isolate it from humans. So this was only a protein as compared to insulin, which you can apply isolate from pigs. EPO has always been made recombinantly in E. coli, not in E. coli, actually, I should say, in cell lines. And that is because this glycosylation is very important. And some of you might know glycosylation is different in different species. So you can't actually just isolate EPO from pigs and inject it into humans. That's not really possible. So really developing this is only possible because of recombinant DNA techniques. It was first approved by the FDA in 1989 for therapeutic use. And you can see it's actually off patent already in itself. But glycoscience has always played a very major role in developing EPO. What Amgen managed to do is obviously, well, make a big profit from EPO. But also what they managed to do is try to understand the glycosylation of these proteins. And once they understood what it looked like and what the function was and so on, they managed to actually manipulate it a little bit, and in fact actually managed to get a sort of second generation EPO onto the market, which is Aranesp, which has a longer half-life, just purely by manipulating actually the carbohydrates. They introduced actually another glycosylation site, put more carbohydrates on, which made it stable, which gave it longer half-life, and actually they got extended their patent lifetime as well. So it made them a very massive profit understanding, what the glycosylation of EPO actually does. And as I said, it's still growing. I think we are now at a stage, and this is again makes this very interesting for other people other than Amgen, is that we are starting to look at generic drugs now. These things are coming off patent, and you find that there are a lot of companies interested in developing generic drugs, and you certainly need to understand the glycosylation to prepare the generic drugs. So this is a very, very big area of research. And following EPO, for a long time, EPO was the only glycoprotein on the market. All the other protein pharmaceuticals were non-glycosylated. Insulin is not glycosylated. But actually, over the last 10, 20 years, a lot of therapeutic antibodies have gone onto the market. They have a different sort of glycosylation pattern. The carbohydrate has a slightly different role, but is, again, very important. And now the top-selling drugs are all glycoproteins, or most of them are glycoproteins. I think 8 out of the 10 top-selling ones are glycoproteins. So really studying this glycosylation is very, very important. So what do these carbohydrates on the cell surface look like? Let's dig a little bit more into the structure. I do appreciate that you're not all chemists. If I showed you the chemical structures, they look really like sort of big forests of structures. And so what we tend to do in glycobiology, we tend to use symbols for the different monosaccharide building blocks, just to make these structures a little bit more accessible to people. And if you're interested in glycobiology and you want to learn more about it, I can highly recommend this textbook here, Essentials of Glycobiology. It's an online book. I think there's a new version coming out fairly soon. And that gives you sort of a really nice overview on glycobiology if anyone is interested. It's all online, freely available. You can buy it as a book, but you can just get it easily online. And that's a really, really good book to introduce you into glycobiology. It has very nice introductory chapters. And some of this slide comes from that book, I should say. So you've got this sort of cell membrane. I said before, you've got this glycocalyx, and you can see just by looking at the symbols, so for each sugar, monosaccharide building blocks, we use a different colored symbol. And I should say, maybe I'll just go to the next slide for a minute. I should say also in this Essentials of Glycobiology book, there's instructions on which symbols to use. When you generally use symbols for polymers, you just choose them yourself, whatever looks nice in terms of color. But actually, we're now trying to encourage people, and this has not been adhered to in the literature in the past. If you look at the old literature, people use all sorts of symbols for carbohydrates. And actually, it was a real big problem to convince the whole community to sit down and say, right, which symbols should we use so that we can communicate? That you don't get a slide where people use red, blue boxes for glucose, and the next talk uses a purple box for glucose, so that we all use the same symbol. It took actually a long time, surprisingly long. Now, anyhow, very recently, there's this paper now where the whole community has pretty much decided on what symbols to use. So if you work with carbohydrates, and this is actually probably not even for those who work on carbohydrates well known, I really encourage you to sort of look this up. This is all again on the essentials of glycobiology. This slide you can download from the internet. And so you have to be quite precise on this. These are not sort of Mickey Mouse symbols. So galactose is always a yellow circle, and glucose is always a blue circle, mannose a green circle. This only works obviously for the main building blocks. If you have an unusual one, you use a letter inside a symbol, and so on and so on. So there are rules now for representation. So please, we also then signify the linkages. Obviously, if you have a simple polysaccharide like glucose, sort of like cellulose, you just have blue circles, regular blue circles. And I think it's a very nice visual way of presenting carbohydrates. Because if I show you a cell surface glycan and a cellulose, and you're not a carbohydrate chemist, you sort of wouldn't see the difference really very easily. Whereas this really quite quickly in one look can show you what to do. So this is a very recent development in glycobiology, and as I said, if you work with sugars, I strongly recommend you use that right from the beginning. There's also software which allows you to build this as a glycan builder software and so on. So we've been trying to really make these structures more accessible to the general community, and also to biologists in particular, who are not sort of structural people. So that's how we present them. This is sort of almost equivalent to using sort of ribbons in presenting proteins. Yeah. Right, so let's go back to these. So you can see they're not very homogeneous. They contain, the way they're presented here, these are the main classes. They actually contain quite a small set of building blocks. If you remember, with amino acids, we have 20 amino acids naturally occurring. Generally, the sort of fundamental, most glycans are made up of quite a small set, surprisingly small set of building blocks. However, they're not limited to that. So I wouldn't be able to write down all the sugars, that are available in nature. It's a massive number. But what you have to understand is biology only uses a subset. Okay, so biology doesn't use absolutely every sugar that there is. In some circumstances, if you get an odd organism, you might have a funny cell surface carbohydrate, but generally they're limiting themselves to sort of a core set for sort of basic function. And despite the fact that they're so diverse, you can see you've got linear ones here, hyaluronan, chondroitin sulfate, heparan sulfate. Some of you might have come across. It's a highly sulfated polysaccharide, but it's also sort of a long chain. Tends to be longer. Then you have branch structures, and this is meant to present a protein. So what I showed you in EPO as a coarse protein structure is this, and these are now the symbols of the carbohydrates linked to these proteins. And you can see they're branched here, so they can be branch structures, and you can see also they're not made of the same building blocks. So this is a so-called N-glycan, or one N-glycan structure, and they've got blue boxes, N-acetylglucosamine, and green circles, which is mannose, and then they can be further decorated with phosphates or sulfates. So S means sulfates and so forth. They can have fucose here. This is another sugar. Sialic acid is a purple lozenge, sort of diamond type of structures. You can see they're quite diverse. They're not like cellulose or starch, where you just have this glucose as a building block. However, again, they use a limited set of building blocks, sort of shown here in this box, and also they have actually some conserved structures. So again, not every variation of carbohydrate is used in biology, and the conserved structures, highly conserved actually between species, surprisingly, are these green boxes here. So this is a very, very famous core pentasaccharides of N-glycans. So N-glycans are all the sugars that are linked to asparagine residues. So they're only linked to asparagine residues in protein, or mainly, very, very rare exceptions. So always asparagine. Only one of the 20 amino acids is picked as a glycosylation site, and that's what they're called N-glycans, but they're linked to asparagine. They have, and they're almost pretty much throughout king have this core pentasaccharide structure, certainly in higher organism. This core pentasaccharide structure that comes through the biosynthesis, which I can't really cover here. So there are these these sort of core structures. Then we have the O-glycans. These are linked to serines and threonines. Again, picking just mainly serines or threonine. Again, sometimes you can get other exceptions. And they would either have a mannose or an N-acetyl-galactosamine as the first sugar, always that. Some of you, I know, here study glycnacylation. We call it glycnacylation because this is adding of N-acetyl-glucosamine. So this is an extra, sort of intracellular. So this is the extra. This is the intracellular, and this is an intracellular glycosylation, and that's just adding one acetylglucosamine onto proteins. And this is sort of a process that sort of competes with phosphorylation. So a lot of people are very interested in this, regulates protein function and so on, but a little bit like phosphorylation. So a lot of people are interested in the enzyme that puts that on and cleaves that off. So, and then obviously you've got the lipids here, so glycosphingolipids, GPI anchors with their very specific structure, which are actually glycolipids that anchor proteins onto cell surfaces. So these are all these cell surface proteins, carbohydrates that are now sort of presented there. What I'll be talking a lot about is side chains of glycoproteins in this. So just to sort of focus yourself in, I'm not going to talk too much about these, very interesting class of carbohydrates as well, but I'm focusing a lot on these sort of structures, the side chains of glycoproteins, the ones you find on EPO and catalytic antibodies. So a lot of my examples will come from that. Okay, and as I said, these are the structures. And the first thing I want to do, how do we actually determine the structure of these? How do we do glycomics? The first issue with glycomics is to actually find out what carbohydrates are in a cell or organism. How does that compare to other cells and organisms? One person has one glycome. How does that differ from the glycome of another person? You have two cell lines. One is a cancer cell line, one is a normal cell line. How does the glycosylation differ on that? And what we're all looking for is obviously biomarkers, which is actually a big issue, and I'll show you some example of that. So for that, we have to have fast sequencing methods. And this slide is just to remind you that sequencing is always incredibly useful in driving science forward, and we've all seen that with nucleic acid sequencing. The whole revolution, DNA revolution, will only be possible with cheap sequencing methods, and we'd already covered those in lecture 2, of course. So this is a slide I'd shown in lecture 2. That sequencing cost has come down enormously. We can now very quickly determine the genome of a human being. We can't do that with a glycome at all. We're way behind with the sequencing methods for glycomics. So that's sort of a huge gap, technology gap, that at the moment we're trying to fill. And there's quite a lot of effort in science to really develop carbohydrate sequencing method. And that's what I sort of talk about today in my lecture. How can we actually determine the structure and the sequences of these carbohydrates? And why is it so difficult? It's good to compare the different sequencing methods. This is DNA. This is quite a complicated molecule, in fact, a very large molecule. These are proteins. So you only have got 4 building blocks there, of course, and you're limited to 4 building blocks. Proteins, you have 20 building blocks. And again, structural diversity is quite, it's quite a complex molecule. And then you've got these carbohydrates, which are actually often a little bit smaller. Why is it so difficult to sequence these? And this is easy. Well, with DNA, this has a very, very regular structure and only 4 building blocks. So we saw all these very elegant Sanger sequencing methods that had been developed. So you can develop very specific sequencing. You don't actually need to know very much about, whether this ribose oxygen points downwards or upwards, about the stereochemistry. All the building blocks are very, very homogeneous. There's only a very limited set of building blocks, and all you need to know is what do you have there. Do you have a T, an A, G, or C? So that makes that easier. Very dedicated methods. You can't really apply those methods. You can't even apply them to proteins or carbohydrates because you've got far more building blocks and far more complexity. However, proteins, you've got 20 building blocks. You don't have a polymerase or anything like that. So the Sanger sequencing type doesn't work, which I introduced to you. So what do people use for protein sequencing? Well, there are lots of methods available. I can't go through, but one method, which is nowadays mostly used for protein sequencing, is mass spectrometry. So basically, if any one of you has done some protein sequence determination, what you do is you put these proteins, you fragment them into peptides, and then you chop them up in the mass spectrometer, and you just basically measure the mass of each building block. That's by far the best way nowadays to get a protein sequence. That's what it's generally done nowadays. So mass spectrometry now starting to creep in as a very high throughput sequencing tool, and it's been tremendously successful for proteins. So I mean, advantage with proteins is always that we can that they relate so closely in sequence to carbohydrates, so we can always just do sequencing, DNA sequencing. But if you have to really identify a protein nowadays, it's actually quite straightforward to get the sequence out, generally. And that's through mass spectrometry, and I'll come back to that later on. So mass spectrometry, very important for sequencing. Now, why is carbohydrates even more complex than proteins? I've listed here a few reasons. For example, the structures can be branched. They can be linear, as I've shown you before, but they also can be branched, and that obviously makes sequencing, you can imagine, a little bit more difficult, because sequencing generally relies on just chopping up the chain and just going step by step. Carbohydrates are, with the branch structure, it's much more difficult to design. You don't know how branched they are. Some are branched, some not, where the branch is, and so on, makes it all more tricky. The building blocks are isobaric. So I said already, so isobaric means a lot of them have the same mass. I said already, what we're using is mass spectrometry often to sequence proteins, and very few of the amino acids have the same mass. Okay, I mean, leucine, isoleucine will have the same mass, so you can't distinguish by mass spectrometry between those two. But most of the 20 amino acids have different mass. So you can identify them in the mass spectra just by their mass, the building blocks. So if you chop them up and you take off one amino acids and it's a certain mass, you can immediately say which amino acid it is. You can't do that with carbohydrates because, first of all, they're more diverse, but also, if you look at this trisaccharide here, for example, which has mannose, glucose, and galactose, and you look at the structures, each building block has got the same number of atoms of each type of atoms and number of atoms. So they would, in a mass spectrometry, they would all look exactly the same. Yeah, so they're isobaric. So you can't use pure mass spectrometry really to sequence them. So that's very difficult. And they're anyhow very similar in structure. If they had different structural features, you could, if one had an acid and the other one had a base, you could try to do some analysis on this. But if you look at these again, and I should say, these are very, very different sugars, have very different roles in biology. If you go look at glucose, which is the most common sugar, if you look at this as this ring form here, which sits in this what we call the chair form, it's not very important here, all the oxygens around there and all the substituents point into the plane of the chair, So it's very characteristic of glucose. It's a very stable sugar. All the substituents are pointing away from this chair. If you then go to mannose, all you've done is taken one of those hydroxyl groups and flipped it up. And everything is exactly the same. Yeah, so same composition, just changed one stereoisomer here. Totally different biological function, mannose, but it is but it's structurally, chemically incredibly similar. As a chemist, this is the beauty of nature, that nature does makes very simple changes, and in fact, actually biosynthetically, these two are linked. There's an enzyme that converts glucose into mannose. Yeah? So in order to generate a new sugar, nature also only has to put in this enzyme, swap over the stereochemistry, not much effort, get some completely new biomolecule which they can use for something completely different. And equally galactose, if you go from glucose, it's just got this position here flipped over. Yeah? So they're really, really similar structurally, and very difficult to find physical methods that distinguish between those. Mass spectrometry, which is generally used for sequencing, certainly can't do that. So they're very similar in structure. Then the other thing is, if you look at the linkage, so again, the big linkages in biology, phosphodiester linkages, I've already talked a lot about that in my last lecture. You've got the amide bond here, so that links all the amino acids. So that's the big linkage used for amides. Similarly, ester, if you're an oxygen here, that's used for lipids. For sugars, we have the so-called glycosidic linkage. All sugars are linked to other biomolecules or to themselves through this glycosidic linkage. So what's the glycosidic linkage? So it's basically this linkage that is not always, but often is the sort of the last carbon or the first carbon. It's normally numbered as C1 in a sugar, in this ring, the carbon next to the oxygen, and it's linked to two oxygens here. This is what we chemistry call an acetal, and that's actually very susceptible to hydrolysis. So it's again a linkage, like all the other linkages, that can be quite easily hydrolyzed with water. So that's what you want in biology, linkages that are made through condensation, removal of water, or adding water. They're always a good linkage. So in that sense, you know, if you cleave the linkage, you have to do hydrolysis, very much like you have to do when you cleave these. But these linkages are quite different. And again, they have some complex chemical features, much more complex than these linkages, which again make them quite difficult to analyze, but also are very important for their biological role. So if you look at these two centers here, never mind you have different building blocks, this oxygen points downwards, we call that an alpha linkage in this particular sugar, and this one points upwards, and we call that a beta linkage. And it's very important you get alpha and beta right. So the difference between cellulose and starch, so starch is something you find in rice and potatoes you can eat, cellulose you can't digest. Difference is just that cellulose has a beta linkage and starch has an alpha linkage. It's just purely the only difference, and that it makes it incredibly different in terms of, well, actually in terms of chemistry, cellulose is much more stable, as you all know, but also in terms of biology, you know, the enzymes that can degrade cellulose and enzymes amylose, and they're not interchangeable. So it's very important, if you determine the structure, that you also determine whether it's an alpha-beta linkage you have. And then the other problem is that you have got not only one linkage point, you've got actually several linkage points on each sugar. Here, with these, you've got only one acid and one amine. Here, you've got the phosphate is always linked to the 5' and 3' hydroxyl group. In fact, in DNA, you don't really have another option anyway. But in sugars, you've got all these different hydroxyl groups. They're all, again, very similar in structure and activity, and you can link it to different linkages. So what we do is generally we count around a sugar 1 to 6, and we then say which two carbons are linked to each other. So if this is the 1 carbon linked to the 4 carbon, here the 1 carbon is linked to the 6 carbon, and so on. So again, you know, that is really important that you determine that. So the linkage contains stereochemical information, and several linkages are possible. So what we need to do is actually, when we determine the sequence, we need to look at the stereochemistry. You know, look at you could call it three-dimensional shape. Yeah, so it's not good enough to just say, okay, there's a hexose here, another hexose, and another hexose, which would be fine with these because there's no other option. You can only get one linkage. With sugars, you need to really dig into this and do a much more thorough analysis, and that makes carbohydrate sequencing so difficult. So how do we how do we do it then? So the classical way, and this is the gold standard, and this is how most carbohydrate structures actually have been determined so far, is nuclear magnetic resonance spectroscopy, NMR. So this is shown here, a picture found on the internet by a Bruker instrument. And really what people have to do is they have to get a pure sample. So, you know, polysaccharides or protein side chains, they're all laboriously been isolated by scientists in the past. You need milligram quantities, which is quite a lot often for biological material. And then if you have that, you tend to get actually quite complex data, and the elucidation of these data can be quite time consuming as well. But there has been there have been a lot of advances made in the field in terms of structural elucidation. And in particular, a colleague of mine, Göran Wittmann, has developed an automated sort of assignment system for carbohydrate called CASPER, which is described in here as an automated shift prediction for carbohydrate, which makes the elucidation somehow more easy. So this paper here describes this example where he does a carbon-13 analysis of a carbohydrate. He gets all these different, very individual shifts, chemical shifts. What you get in NMR is chemical shifts for each atom. So that's the beauty about NMR. So each carbon in your carbohydrate has a different chemical shift. You can sort of catalog that. So these are the chemical shifts in the carbon-13 spectrum of this polysaccharide here, which he worked on. And you can see here again specific linkages, specific building blocks, some branching and so on. And he demonstrated in this paper, here's a fairly recent paper, that this program CASPER could actually come up with some very viable proposals on what the structure should be. So NMR is getting more and more advanced, and NMR, as I said, remains the gold standard. But if you want to do glycomics, if you want to look, in the clinic at all the carbohydrates in a cell or something, NMR is just not feasible. So NMR is very, very good to set the standards, to really fully characterize, unambiguously characterize structures. But for high-throughput biology, to catch up really with genomics and proteomics, NMR is really not very good. And as I said, one of the big problems is that you need large amounts. For mass spec, you need very, very little. For NMR, you need quite large amounts, and you need pure samples. But as I said, it's very, very important. I'm not going through the sort of theory of NMR here, so there's not the time, but I think I mentioned that. But as I said, I want to talk a little bit more about mass spectrometry. I know you do mass spectrometry here as well for structural elucidation and sort of limitations in where we are and where we think we're going to get actually stereochemical information from mass spectrometry. This is just another sort of way of explaining, all this stereoisomerism, the complexity of the stereoisomers. As I said, you have got different building blocks, mannose, glucose, galactose. You've got alpha, beta anomers. You've got these regioisomers. How do you determine that? If you do a mass spectrometry, you can actually get quite a lot of structural information, and this is very similar. So basically what you do, let's say, look at a disaccharide, just to make it all quite simple. What you can do by mass spectrometry, you can obviously determine the molecular weight of that disaccharide, so that gives you already some information. You can actually do the so-called MS-MS techniques, whereby you put it through a mass spectrometer and then you fragment this compound, and you get a very characteristic fragmentation pattern. One of the main fragments due to cleavage of the glycosidic linkage. So basically, very nicely, in the mass spectrometry, you actually cleave that glycosidic linkage. And in peptides, it's the same. You cleave that peptide linkage. So it's a very nice feature that you can use. It is actually the weakest linkage. This is sort of, for example, using CID, collision-induced dissociation. And then you can just do another mass spectrometry, and these are sort of, we call them tandem or MS to the power n mass spectrometry, where you just send the molecules through a series of sort of mass spectrometers, and you can manipulate the ions that come out of the first mass spectrometer and send it into the next one, for example, by dissociating them. And obviously that gives you even better information, because now you get the so-called B ions and C ions, depending on where this linkage breaks. It can break to the left and to the right of the glycosidic linkage, and again, that's nice additional information. Now you can determine the mass of this, and let's say you can say it's a hexose, and that narrows down, of course, tremendously what sugar you have. You don't have a pentose or smaller molecular weight sugar. If you do this carefully, you can even look at some modifications like methylations, for example. You can distinguish an acetyl glucosamine from a glucose. So this gives you enormous information. Also, you can already tell what order they're in, actually, with various techniques. but it does not tell you these stereochemical. It gives you information. So if you take any of these two, for example, will give you exactly the same mass spectrometer, sort of shown here a little bit. If you look at, for example, GLIC beta-1,4-GLICNAC and GAL beta-1,4-GLICNAC, they will give you exactly the same mass spectrometer. Maybe a little bit of difference of height of peaks, but you really can't really interpret structures on that basis. So very, very useful for getting sort of, well, I call it two-dimensional structure. You have one of these, or hexos, one of many. I mean, there are only a set number of hexos you can have, but you don't really know which one. So how do you get more information out of this? So no stereochemical information. So you just need additional protocols in line with your mass spectrometry analysis. And there's been, again, a massive amount of work done. So, and these are very complicated protocols, and I'll go a little bit through this. One of the protocols involved, and I know that's very much used here here as well, is developed by Pauline Rudd. Originally, she was at Oxford in the Glycobiology Institute, but she then moved to the Glycobiology Institute to Ireland, Dublin, in Nibud, and Professor Liu was actually a postdoc in her lab. So this is a recent paper from them, but they've published many, many papers and really optimized this protocol. You can see it's quite complicated, sort of at first sight, but there's actually now a commercial instrument available that actually allows you to do this particular analysis in a sort of fairly automated fashion. And in addition, the Rudd lab has used it actually to get some very interesting data on biomarkers, for example, and I'll show you them in a minute. So what do you need to do to really dig down into these three-dimensional structures? This is sort of the overall workflow of protocol, and some of it is actually very similar to maybe also things you need for gene sequencing and protein sequencing. For example, you need to get your sample and somehow immobilize it or prepare it. So this is glyco. So I should say this is a fantastic technique, but it's actually very limited to glycoproteins. So this is really very suitable for glycoproteins, and I'll talk about limitations in a minute. So you take your glycoprotein and put it into a 96-well plate format, allow you to deal with many samples. Then you need to get the glycans of the protein. So you want to analyze what glycans are on the protein. And you do this with an enzyme, which again, in Professor Vogelmeier's group here, I think some of you will be working with this, called PNGase-F. Very expensive enzyme actually, so it's good to have good expression system. And what that does, it just cleaves all these N-glycans. You remember there's this conserved pentasaccharide core, always linked to an asparagine, and there's actually an enzyme which generically will always cleave that off. So that's a very useful enzyme, and because the core is so conserved, you can cleave most glycans off there. So this is PNGSF, hydrolyzes very specifically because it's an enzyme, that linkage. You've got very small amounts of material, obviously, so fluorescence detection is the best. So what they do is they label the end, which is we call it the reducing end, has very selective chemical reactivity with 2AB, so that's just a fluorescent label. And then you have actually a whole set of these sugars. I should say these are shown in the sort of old denotation. It shows you, if you go back to the literature, people use all sorts of symbols. So this is just a different way of labeling glycans. It doesn't use color, still uses sort of symbols, but they mean something slightly different. This is exactly what I was saying, happens if you go back to the literature. People use all different symbols here. So I copied that slide here from this publication. So now you've got your set of fluorescently labeled probes. You need to analyze them. And as I said before, mass spectrometry only helps you in a limited amount, in the sense that it tells you about the branching and it tells you all the molecular weight of each building block. It doesn't give you sort of the identity. So what they use is they actually use LC methods. Okay, So it turns out that if you have these isomers, if you shift a hydroxyl group around or shift a linkage around, your LC retention pattern changes. And if you have a whole set of these glycans coming from some biological samples where you get actually a whole mixture of them labeled, and you run them on LC, you just basically get sort of these forests of peaks. And this is highly optimized. So this is HPLC. You can see UPLC. You get really, really nice sharp peaks. And you need that for this type of analysis because you have got lots and lots of samples and you want to compare samples and you want to be very precise. So this technique has been totally optimized. But it's particularly useful. There's a Hillick column, hydrophobic interaction column, which is very useful to get these nice. And each peak here will just be a different glycan, fluorescently labeled glycan. Minute amounts, because you have a fluorescent label. So you have that, but obviously that is just a retention time. So it tells you really in the first instance that you've got all these different peaks there, and you've got lots of different structures, but it doesn't really tell you which structure you have. So how do you assign the structure? And again, this is extremely laborious. So one way of doing it, getting some information, is exoglycosidase digest. So you take these samples here and split them up, and then each of them undergoes you're exposed to enzymes that cleave specific sugars off from the end. So you do your sort of hydrolysis with enzymes, and the enzymes have a certain specificity, and if a certain glycan gets hydrolyzed by a sialidase here, which is this one here, then there's a sialic acid at the end. If it's by a glucosidase, it's a glucose and so on. You can imagine quite a tedious process, but very effective. You can see that simplifies the pattern because you cleave sugars off specifically, and you can sort of to some extent read the sequence by just virtue of the fact that for each step what the sugar is you've cleaved off. Obviously it doesn't work for every sugar because you don't have enough enzymes for every sugar, but it's for common sugars and for N-glycans for which this protocol was designed. It's very effective and gives you very good information. Some ambiguity, but not. So actually you start knowing what's really sort of under each peak, and then you can combine that with your sort of mass spectrometry. And then in addition, you have a fairly good understanding of the biosynthesis, the sort of structures of N-glycans you have in biology, because again, you can go back to massive amount of biochemistry where people have studied these biosynthetic pathways. So we have a good expectation. We know sort of roughly what sort of structures we would expect from these sort of samples. And this whole knowledge of the biosynthesis, the mass spectrometry, the glycosidase digestion, these sort of patterns, you put actually into databases. You can imagine this is an enormous amount of information. So they've generated databases, they quantified all of these. These are all accessible now to people, and also are starting to become accessible to really sort of more general glycoscientists as well. And that allows you now to sort of just assign these structures of each of these peaks. And then obviously once you've done this for one protein, if you go for another protein in the same organism, you sort of start developing an understanding of where these peaks run. If you run your UPLC very carefully, the same glycan will appear at the same sort of retention time. You can compare it with standards, and so you can, after basically, there's now a massive amount of knowledge. There are databases which can allow you to predict retention times or give you retention times for these structures. So, once all this massive effort has been made, and it has been made now and is put into these databases, we can now look at these profiles and then very quickly assign, almost in not automated fashion, but reasonably well. So what experiments are now possible then? I haven't really shown the exact data, but this is a recent paper by them in glycobiology for 2008, not quite that recent. I think it was the best slide I could identify. So we call this Hillick technology, based on this Hillick column, which is key. So you take your samples from serum or from protein, run it down this Hillick column, put them through that protocol. As I said, you come up with these sort of peaks here. I think because this was 2008, this was still HPLC, so they're a little bit broader than the idealized ones I just shown you. And what they did here is they actually looked at whole serum from breast cancer. And basically they had controls, which are the squares, and then breast cancer, which are the samples in black dots. And they basically analyzed the glycans in all these in each of these cell lines, and they can do this because they can use it in 96-well format. And they found that they can actually group the patients quite well through this. This is least square discriminant analysis. So these are just taking all the parameters, putting them into a program, and you can see there's a sort of a very convincing separation between the control samples and the breast cancer samples. So this might be a very good way, of analyzing a tissue in terms of whether someone has breast cancer or not. And that was, as I said, published in this. And you can see actually what they did, what this was all based on is difference of glycosylation levels in these cell lines. And what they're looking at were sort of structures here that are really sort of almost in the noise. But I think this analysis is now so precise, together with sort of some statistical analysis, it really can generate data like that. And there are many, many examples, especially by the Rudd group, where they now have analyzed different cancer forms, ovarian cancer and so on, and shown that actually the glycosylation pattern, and this is something I said at the beginning, of cells changes during cancer. And glycosylation on cancer cells has always been used as a diagnostic tool. People have stained cancer cells. But this really points to the future and shows that glycomics can play a very important role in development of biomarkers for cancer. But this is very much at its infancy. It's really sort of just showing that there's a difference. What they also found, I should say, is there's a big difference between different people and how old you are, whether you're woman or man, and so on. So you have to be very careful with these biomarkers that you don't really sort of mix up sort of natural diversity with diversity due to cancer. But certainly in cancer, I think the glycosylation pattern on these cells really, really changes, and analysis of glycans might provide good biomarkers. There are other methods, sort of using similar, but also using mass spectrometry. So this is just the second method I just want to introduce. Unfortunately, this comes from the Dell group at Imperial College in London. And what they are sort of the leading group in the world, really, to analyze glycans by MALDI-TOF mass spectrometry. So what they do is they don't really derivatize, apart from methylation. So they often permethylate the samples, which it doesn't really sort of show up here, but it makes again, they have protocols in how they do this. So if you just then do just a MALDI mass spec, again, of a whole mixture of glycans, what we do have here is we don't have a LC method, we've just got a mass spectrum. So this is mass, M over Z, so mass over charge on the MALDI. This is the intensity. If you have a mixture of glycans, and I should say, I'm not sure whether I've stressed this, when you look at a glycoprotein, and this is actually the issue, if you look at the glycoproteins, it's never homogeneous, okay? The single sample of, let's say, EPO, will always have a whole mixture of different glycans. You get a lot of heterogeneity on each protein in terms of glycosylation. And I don't really want to go into it, which makes the analysis very, very, very difficult. So I think here this comes truly again from cells rather than single proteins. But you can see if you cleave again with PNGase F, all the glycans off, you get this sort of forest of peaks. And of course, because they're different size, so this one here is, you can see, smaller than this here. You can just see this by looking, at the simples. So large ones are on the right, small ones are on the left in this. Just by virtue of the fact that they all have a different total mass, you can separate them. You can start making predictions on what sort of structures you might have. And again, they have also dedicated programs which people can use. Doing this by hand would be incredibly tedious, as you can imagine, trying to find out what, I mean, all you see, I should stress, is this method doesn't give you that, it just gives you a mass, and you have to make a prediction on what that mass could be. So you can imagine with these masses, with these sorts of molecules, the permutations are just infinite. So when people had to do this manually, that took a long, long time. Nowadays you can do this all with databases. You put that mass into a database and it comes up with predictions on what it could be. And again, because we're dealing with this very limited set of glycans, they're only N-glycans. We know that because we've cleaved them with PNGASF off proteins. We know they're only N-glycans. We know we only can get certain structures in N-glycans. We've got a lot of information on the biosynthesis. Knowledge of the biosynthesis is incredibly important for this. So it's not de novo sequencing per se. It's, it incorporates a lot of assumptions in there. And then what they do is they just basically take one of these and do these tandem mass spectrometry, or MS to the power N, mass spectrometry. So they can isolate this peak in the first mass spectrum, they can fragment it, and then they can sort of do sequencing and find that, they have got this sort of branching pattern and hexanags and hexoses wherever. They use the biosynthetic knowledge, they use the overall mass, put all this information together, and then they can go back to the original spectrum and then sort of just assign it as here. And as I said, this is all done sort of almost automated or semi-automated, in a semi-automated fashion. And that's really nice, as you can see. You can take cell extracts and just look at all the glycans that are there. I should say the height of the peaks themselves don't correspond to the amounts. One thing about mass spectrometry compared to LC is that you don't get a very good sort of correlation in terms of response factors. So some of these might be minute amounts, but they show up very well in the mass spectrometers. Something you have to be careful with if you use mass spectrometry. The size of the peak is not directly correlated to the amount, and changes very much from experiment to experiment. It's not like LC in that case. So the previous method could actually quantify things. Here that's quite a bit more difficult. And just to show you then sort of what experiments they can do, and this comes out of also from a 2008, not so recent paper, so they get a sort of different analysis here. You might, for example, then want to take two different cell lines. So these are two mouse cell lines. One has CD25, sort of one of these immune sort of molecules, and one doesn't. And what they wanted to do is really characterizing the glycome of mammalian immune systems. So they wanted to show what happens when you knock out a particular protein. How does the glycosylation change? And actually, generally, what people find, a glycosylation actually changes quite a lot if you do something sort of pretty, if you often knock out one gene. It almost looks like the glycosylation pattern is sort of almost the most sensitive part of a cell in terms of structural variation. You only knock out one gene, but you change the glycosylations quite dramatically. And at first sight, you can immediately see that there's quite a difference. Not necessarily the position of the peak. So this is the same cell line, just one of this is a knockout. And you can see that all of them can be very easily assigned, and then you can see that you get many more of these higher glycans. With high molecular weight glycans, you suddenly start seeing them, which you don't see so many up there. And in this particular experiment, the conclusion was that actually they get very similar glycans, but at different levels. I should say, relatively, you can make some sort of assessment of the relative quantity of the peaks, not absolute quantities. So lots and lots of experiments like this is now possible, and to some extent, it's a very successful carbohydrate sequencing strategies. And as I said, there are now, fortunately, and this is a massive amount of effort which you all can tap into, because these are all freely available. There have been a massive bioinformatics efforts with international collaborations, very much led by a guy called Klaus Wilhelm von der Lies out of Heidelberg in Germany. He had sort of the lab that was dealing with all these data, combining all the data. Very sadly, he died about 5 years ago of cancer. And since then, it's been quite difficult to coordinate international efforts. But there are these, for example, this database here, EuroCarbDB, is really trying to get all the databases of all the labs, all these sort of efforts I've shown previously, from the Rudd lab, from the Dell lab, and so on, onto one database, so that it's accessible, easily accessible to people. And they've organized that very well. They integrate other databases and PubMed, the protein databases, into this. So, so I really recommend, if you're interested, use that. It should be really easily accessible to everyone. And this database really helps you with, for example, assigning mass spec peaks. So if you can take a tissue, you take a moldy spectrum, you get a forest of peaks, you want to know what glycans they might be due to, you can go into these databases, put it in, and they come up with suggestions. You don't have to, calculate the molecular masses yourself. This is very useful. The CASPR database is linked to it, the NMR database, the HPLC database from the Rudd lab, so that gives you retention times of certain AB labeled glycans compared to glucose ladder and so on. So it's actually some really beautiful effort of a lot of people that put together, and this gets better and better. And I've been in this area for 20 years. When we started, it was just absolutely terrible. Everything had to be done by hand. There were no databases. People were getting results and couldn't really communicate them. Nowadays, people have appreciated, I guess, from success of the nucleic acid and protein databases, that we really need to generate something like this to make it openly accessible. And they've really only just come online. You can see this is a 2011 paper. So people just only starting to generate these databases. They're not quite firmly established as a protein databases, unfortunately, but there's certainly a lot of will to do that. So if you look at the current state of the art of carbohydrate sequencing, then this is, as I said, it's not our work, but sort of the stuff I've been talking about is that really mass spectrometry I think is the way to go, combined with some an ancillary method, because it really requires only small amounts. NMR is fantastic, but really requires too large amounts for serious sort of high throughput analysis, although it will always remain the gold standard if you really want to nail down a structure. NMR is fantastic. If you have enough material, NMR is fantastic. But mass spec is really something we should be thinking about, because it's really high throughput. It can deal with heterogeneity. You just can take cell extracts, isolate the proteins, treat with PNGSF, have all this forest of different peaks. No way you can determine that by NMR. And it's highly suitable for carbohydrate sequencing. But it only provides what I call two-dimensional structure. It doesn't give us this stereochemical information. So it's blind to what I say, blind to three-dimensional structure. You can't get a stereochemistry out of mass spec, which is a really big drawback at the moment. So what we need to do at the moment is combine LC, and for that you need standards, because LC per se doesn't give you any extra information. It just tells you there's this heterogeneity there. Knowledge of biosynthetic pathways is incredibly important. So all these methods work very well for the N-glycans, but they don't really work very well for all glycans. So really, the methodology we have is pretty robust and good for N and O glycans if you work with those. And I should say also the standard ones. If you look at a strange modification, the whole system, might not be so good. So you have to be aware of the limitations. So it's quite focused and limited, driven actually by the biopharmaceuticals. So those people really need good analysis. Obviously, if you put a drug into patients, you need to analyze it. So I think this is here to stay, and it's very, very good if you want to go on glycans. But if you want to do something else, if you've got another carbohydrate, it's not necessarily for the standard NO glycan, it becomes quite difficult, So you really need to go back to NMR then. So what I would conclude is, at the moment we don't have a universal sequencing methods for carbohydrates. And this is, and again, this sort of illustrates the same thing. This is this two-dimensional versus three-dimensional. I'm trying to illustrate this message this sort of information we can get, the branching, but we can't tell which carbohydrates, which linkage, and so on. So knowledge of biosynthetic pathway, then we can get this sort of three-dimensional structure, and we have computation methods to put this back. So it's sort of, I call it shotgun sequencing. Yeah, you break this up through mass spectrometry techniques, you determine the structure of these individual ones with all this bit, and then put this all together. But there is this gap in the middle, and for every glycan, we can't possibly determine the biosynthetic pathway. So how do we get over that? So what we need to really do to address this, what we want to do is find additional methods that give us three-dimensional information, but fast methods. We want to use the advantage of the fact that you get a mass spectrum in a few seconds. You don't want to then run a Hillig column for an hour or something and analyze your sample. You really want to have something that is on the same time scale as mass spectrometry, so i.e. a gas phase technique, really. So can we find some other techniques that can add on and give us that information? So this comes really sort of to some of our research we've done in the past. So I'll spend about 20 minutes telling you a little bit about how we're trying to tackle this. And I should say, I'm not a mass spectrometrist. I find mass spectrometry really helpful, and I think it is a useful technique, actually, also for non-mass spectrometrists, fortunately. But I have actually colleagues in my institute, Claire Ayers, Perdita Barron, who are mass spectrometrists. I'm very, very fortunate that we have a mass spectrometry unit in my institute, and they develop all the techniques, and they don't quite know what to apply it to, so I tell them what they should apply it to, and they develop the techniques. So it's very, very good collaboration, and also helps me get on very well, so it's very nice. And also I've had a really, really good PhD student, Chris Gray, he really needs to mention, who really has been sort of co-supervised by Perdita, Claire, and myself. And he's made up most of the slides I'm going to show you, and he's sort of the expert in this field. So we've been very lucky with having a fantastic PhD student. In fact, most of this work is just one PhD student. He's just absolutely fantastic. So basically, what we did is I was talking to Claire about this and say, look, we need this additional information. She said, well, there is a technique which actually has been used for many, many years in mass spectrometry. It's been around for a long time. It's widely used. In fact, it's used in airports when you get your suitcase swiped for illegal compounds. They swipe it and they put it into a mass spectrometer. It's actually not a mass spectrometer. It's called an ion mobility spectrometer. Okay, it's also a gas phase technique, so also you need to get your sample into a gas phase. But what it does is it also gives you a mass over Z. Sorry, it doesn't give you mass. But what it separates on is not so much on mass, but on shape. Okay, so now we're not looking at shape of biomolecules. So you can imagine some molecules might be very long and thin, some might be very globular. This instrument can actually distinguish between those shapes, and this is exactly what we want. We want to look at three-dimensional shape, and the way it does it is by sending these ions, so these are ions, through an electric field. So they travel through this electric field, and there's sort of different ways in setting up this electric field. This one particular instrument here uses the so-called T wave, but there's sort of other ways of doing it. And then they also have a gas in this tube. Okay, what we call a drift gas, and this could be something like nitrogen or helium, depending on what techniques you use. And as they travel through this electric field, so they don't travel in a vacuum, they travel in a gas, they sort of hit the gas. And depending on their shape, they might be more influenced by hitting the gas than others. So, something that's quite small doesn't hit the gas so readily, something that's quite big hits the drift gas more. And that influences the speed by which they travel through this tube. Okay, so it's a very nice complementary technique, especially if you combine it with M over Z. Gives you the charge of the molecule, the mass of the molecule, and the shape of the molecule. So it's not a new technique. It's now widely used. It's just never been much applied to carbohydrates. And the beauty about it is, what it used to happen is people used to build their own drift tube, but nowadays you don't have to do that. You can actually buy a lot of instruments. I'll show you a few instruments. So basically what these instruments, and I don't want to go through the detail, I wouldn't even be able to explain to you the detail, but what you need to sort of ionize your samples, and then you have got different sort of modules of different techniques linked to each other, and you can buy these integrated instruments. So this instrument we have is a Synapt G2, so by the Waters company. They're outside Manchester, so we have a lot of collaboration with them, so that's very good. And basically, a lot of it looks like a mass spec. This is the TOF, but integrated into it is this ion mobility drift tube, which basically does this. I've just explained. So the question was, can we use this setup, which is totally integrated and is the same time scale as mass spectrometry, to actually separate these isomeric glycans? And I should say, let me just go through, Chris made these slides. In fact, actually, I should say, we've just written a review in Biochemical Biophysica Acta. I think it's just in press at the moment, so it is. It's Gray et al. And he's actually, Chris has mainly written it, and he has put in a lot of these instrumentations in. This is just to convince you don't have to go with one company. And then there are also these different setups now. This is one by Brooker. You can also, I think, buy the individual drift cells and stick them onto your mass spectrometer if you have a... So there are lots of ways of doing it. There are different ways of generating this sort of electric field. So one is called traveling wave. That's the one I've just shown you. This is high field asymmetric trapped ion mobility in B and C, and whatever the last one is called. So they're different conformations. As I said, this technology is not new and has been around for quite a long time. And actually, we're using it increasingly, and I'll talk about it on Saturday as well, for other bioanalytical applications, because it's very insensitive to impurities. So it's a very nice technique to integrate. There's an Agilent instrument. Again, so you see they've just integrated the drift tube into this whole setup. So there are a lot of different things around. So the question was, in fact, I should also say, a lot of this work, this instrument has been used for looking at proteins. There's a very, very successful group at Oxford in Britain, looks at protein shapes, assemblies of proteins. Do proteins assemble in sort of long line, or do they form very globular assemblies? And with this technique, they managed to show differences. Carol Robinson, I don't know whether anyone of you have heard of, but it's a really useful technique to look at shape of biomolecules in general. So the question is, what about carbohydrates? The shape here, as you can imagine, doesn't actually change very much if you go from glucose to galactose, for example. As I said before, this changes hydroxyl group around. You can think, maybe it changes a little bit. And we were just wondering whether it was sensitive enough to actually see a difference. And you can see, actually, we can indeed see a difference. So this is now the time it takes this molecule to go through the drift tube. So this is the drift time. And you can see this is in milliseconds, so very fast techniques. It's not like HPLC. It is actually has got some characteristics of electrophoresis or HPLC. Yeah, so think of it as HPLC in the gas phase. You don't need a complicated setup, and it's very fast. And you can see actually 3.84 and 3.68 is different, and it's significantly different because you're actually dealing with a very defined technique, very defined tube, very defined gas phase. You can actually control the pressure of your drift gas and the flow and everything, temperature and everything. These are very, very precise, and they're very reproducible. Yeah, so these are, we can rerun the experiments and get exactly the same number. The error bars, I haven't put them in here, but they're really much smaller than the difference. Actually, I have to admit, in this particular experiment, difference between mannose and galactose, so these are the ones up to these two up, is actually not significant. So that's not significant. Those ones in that particular experiment we couldn't separate, but glucose and galactose we could. Okay, and then the NACs, the same thing. Actually, Galnac and Mannac in this experiment wasn't very good. but glycnac and galnac, you can see you can separate. And these were actually the first results we obtained showing that you can separate these things and distinguish between these. And that was extremely promising. We published that in Nature Chemistry in 2014. And nice thing is also with this method, in combination with getting experimental data, you can also try to calculate what the drift times might be. And basically what you're looking at, as I said, if these molecules have different shapes, you look at what we call the collisional cross-section. That means, how big is this molecule? The molecule obviously tumbles around a lot. How big is this molecule, and what's the chances it interacts with this drift gas? This can all be calculated much beyond me. But just to sort of illustrate, obviously these molecules are not round, but because they're tumble, they have some sort of average radius around them, and there are various models which people use to calculate them and to try to predict what these collisional cross-sections would be. Because it's a gas phase technique, it's very amenable to calculation. So Chris actually in our lab did calculate these, and these are these collisional cross-section values, and you can see again not much difference between those two, but quite a bit of difference between those two, suggesting that you can separate them. And then these collisional cross-section you can actually also calculate from your experimental data. So you can get these. So these are actually generally the values we look at from the drift times experimentally, but you can also calculate them. So that's also another feature of this. You can sort of try to predict what the collisional cross-sections of these molecules might be. Once we accumulate a lot of data, maybe we'll be able to sort of predict these, and that allows us maybe to avoid having to do massive numbers of measurements. So with this now, we can actually add to tandem mass spectrometry and look at these. And as I said, these are these collisional cross-sections, and they're different we found from glucosamine. So can we resolve isobaric monosaccharides? Yes, we can. You can see actually the difference is not that impressive. If you had LC, you would say, well, that's not particularly good, but it's highly reproducible and really can be used to identify a particular monosaccharide building blocks. Now, why were we actually interested in that? Why do we need this method when we know already what the biosynthetic pathway is? We always know where the glucose and the mannose and the galactose is in glycans. There's no other way. Well, we were very interested in studying enzymes, and I'll talk tomorrow more about glycoenzymes. But we were looking at glycosylation of some peptides using an enzyme called pp-galnac-T2. It's a mucin glycosyltransferase that introduced the first sugar onto peptides. And we were looking at these in vitro, and we were wondering about the substrate specificity of these enzymes. So what we did is we did a screen, and we did this on arrays, and I'll talk tomorrow about arrays as well. Anyhow, basically did a screen of different peptides against different sugar nucleotides, substrates. So basically a screen of two substrates, two-dimensional matrix of screen, and every time you get a positive, plus negative, you get a minus. These enzymes in biochemical journals, if you look them up, are meant to be highly specific for their substrate. Okay, so we would expect actually the substrate is UDP-galnac, that we only get pluses really in this row against a whole bunch of different peptides here. You can see not all peptides work. There's some peptides which didn't work particularly well in this assay. But actually, interestingly, you see actually more pluses than we expected. I should say this was an experiment we could do very quickly. We meant to do it sort of as a proof of concept for this array technology. So when it looks a bit tedious, if you expect lots of minuses, why would you do the experiment? But it was actually an experiment that could be very easily done on a microchip. And it was a great surprise that we saw these pluses. So we thought, well, we've used a lot of unnatural sugars here. These were provided to us by Rob Field in the UK, and some of these are not natural, and maybe the enzyme accepts other sugars that it never encounters in normal life, and that's why. But if you look actually at the hits, these are two sugars, UDP galactose and UDP glycnac, which are actually naturally abundant. And actually UDP-GLICNAC, those of you who know a little bit about sugars, is actually 10 times more abundant than the actual substrates. So that would suggest to us, really, that maybe UDP-GLICNAC is sometimes also incorporated into these mucins. And actually, if you look at the literature, no one has ever reported that. No? So it looks like this enzyme is actually a bit more promiscuous than you might think. Now, obviously, this was in vitro. So we said, okay, let's go in the literature and see whether anyone has found incorporation of this GLICNAC into mucins compared to GalNAc. And of course, no one has described it. And you could say, okay, because people were looking for it but didn't find it. But actually, no one has looked for it. And secondly, there's no technique really available to distinguish between peptides that have GLICNAC and GalNAc, because all the analysis done by mass spectrometry, people learn the rules. This enzyme always puts UDP-GalNAc onto the first peptide, incorporate that biosynthetic knowledge into their sequencing analysis, and then assume if they see a hexNAc, it's UDP-GalNAc. And this sort of shows you that it is a little bit dangerous to use these sequencing protocols, because you just always assume that everything that's in the literature is correct, and it not always is. So that's what really got us into using this ion mobility mass spectrometry. So what we did is say, okay, these peptides, so they're now not even the sugars themselves, but actually the peptides with only one stereocenter difference, can we resolve them with our G2 instrument? And it turned out we can. So these are two MUC mucin peptides, MUC2, and the only difference, these are 14-mes, so quite large peptides. The only difference is really that we have a GLICNAC and there's a GalNAc. Only one hydroxyl group sort of flipped up at the C4 position of the sugar. And you can see it makes the peptide, run differently on this ion mobility, significantly different. You can do statistical analysis. Obviously, they are quite close in drift time, but definitely different. One of them even has a little shoulder on there. Now, again, per se, that is not a good sequencing strategy, because it's just a drift time. So we would have to make standards. But what we can do is tandem mass spectrometry, cleave in the mass spectrometer the sugar off, and then only look at the sugar. So this is this tandem mass spectrometry I've shown before. And now we can easily say, okay, one peptide, the red one, comes from the GLICNAC, and the blue one comes from the GalNAc. So this is a really way of sequencing glycans without doing, which was never possible before, by purely sort of mass spectrometry techniques. So that is very nice. We apply this to more complex molecules. So we said, okay, can we now go back maybe to these N-glycans? As proof of principle, we obviously have good sequencing strategies for this core pentasaccharide. Again, going back to that. But maybe can we use our method to actually get stereochemical information out without relying on the... And then the way we did this is by taking the pentasaccharide and fragmenting it into smaller fragments again, and getting all the drift times and all the collisional cross-sections here. And of course the squares, which have actually different mass, have different drift times as well. But you could easily identify them as N-acetylglucosamines as opposed to galactosamines, and these as mannose as opposed to glucose, and so on. And then we generated sort of a database from other glycans which generate similar fragments. So we used these two, which are commercially available, and fragmented them. And you can see they generate the same fragments. So if this was a structure as we suggested, and this was a structure as we suggested, we should be able to compare these fragments, and they should all match up. Okay. So in a future experiment, what you might want to do if you have a new glycan, you sort of fragment it, measure all the drift times, and then go back to some database which has been accumulated before, and sort of determine the structure of these fragments, go back and determine the structure of your unknown. So this is sort of a proof of principle that this whole sequencing strategy, three-dimensional sequencing strategy, works. And I should say there are now other groups working on this. This is a very nice paper. Actually, we cited it in our BBA, but it's actually by Hoffman et al. In Nature. It's just come out in Nature. where they've actually showed, for example, ion mobility. The same methods can be used to resolve to anomeric structures. So here they don't do any fragmentation, they just look at synthetic sugars, and they can only differ through the anomeric configuration. And you can see that they actually, those two, they get very, very nice separation in these measurements between two stereoisomers. You see very different types, and they can actually then also quantify them because they get baseline separation. So I think this technique of ion mobility integration into mass spectrometry is getting more and more used now by people to look at really detailed stereochemical information of structure. I'll give you one more example before I finish, and that is we worked with some plant people. I thought maybe some people were interested in a very nice group, Berger, Lindberg, Möller, at the Carlsberg laboratories in Copenhagen. So Carlsberg laboratories are the laboratories which are assigned with a brewery. There's a Carlsberg beer which sold, and but the Carlsberg beer company also has a foundation, and they have a very big research lab. So Berger Möller, I think, is at the moment the head of that research lab, but he's actually a plant scientist. So he's actually interested in cyanogenic glycosides from various sources, cassava, but also almonds here. And these are used sort of as defense molecules. So these are glucosides, so you've got a sugar here, and you've got this very specific hydroxynitrile, which upon external stimuli releases HCN, and that's used as a defense. So almonds often can sometimes taste of HCN, so they come from these hydroxynitriles. So they in the plant they are retained. Obviously, they would be maybe toxic to the plant as well as these molecules. And to make them all water soluble, really, they're sort of generally glycosylated. And basically, what happens is if you look at almonds, again by LCMS, and you look at these sort of structures, you find a whole mix of different glycosides, massive amount, really a sort of forest in your HPLC. Berger, Möller and his group have done quite a lot of analysis, and they've managed to sort of identify these structures as these glucosides. There's an APO sometimes there. There could be 1,6 or 1,3 or 1,2. They're all there sort of in the mix. Also at different aglycones here, you can see not only the ones I've just shown before, depending also on where you come from, whether from sorghum or cassava or almond, you get different mixtures and so on. So again, what they needed is a good method to analyze these mixtures, very much akin to what we just seen for N and O glycans. The problem here is we don't know much about the biosynthesis, and I think the idea is that the biosynthesis is not very regulated. There are just some glycosyl transferases there, and it probably depends on the expression of the individual enzymes, the particular day, particularly state of the plant, on what you get there. So we can't really use biosynthetic knowledge here to sort of assign all these structures. We can't say, oh yeah, from almond, we always have a 1,6 linkage here, so we don't need to really look at any other linkages. So this, we thought, was a very beautiful target to use our sequencing strategy to identify their metabolites and see, can we use it now in a real example, in an example where we really need some more information. Also, what we were very quite attracted to is the fact that these are quite small structures. You could start with small structures, So actually, sugar composition, which sugar do we actually have on there, galactose or glucose, we couldn't really do. What linkage type? Of course, if you got enough of one of them, you can do NMR. You can, of course, synthesize all the different standard and compare them by HPLC, but this is all very difficult. You've got different aglycones, you have to make all the different standards with different aglycones, with the different linkages. Really quite complicated. So we thought, we proposed we have this simple sequencing strategy, fast sequencing strategy. Can we help them with the analysis? And we focused really on one particular problem they had. This was just before a publication, that isolated this compound here from almond. And they knew that they had glucose here. They knew there was a beta-1,6 linkage. They knew what the aglycone was. These are called amygdalin glycosides, if anyone knows about these. And they had an LC, so this is an LC trace here. And then they knew this was not homogeneous. This was not one structure. They knew there was a hexose there because they can do LCMS. All these 3 peaks gave them exactly the same LCMS spectrum. MS spectrum. Obviously different LC, because by LC you can distinguish them. But they had no idea what the hexose was, what the linkage was, or whether it was alpha or beta linkage. And obviously if you want to publish these structures, quite tricky. You say, well, that's it. And also, you can see the noise is quite high. They really didn't have enough material to isolate those. So we used all these ion mobility technologies. We had already, because we generated a lot of standards, I hadn't shown really the structure, we had all the data on fragments we can obtain if you had a mannose there, if you had an alpha mannose or beta mannose there, or galactose or glucose, all the possible hexoses. We had standards, very simple standards from other experiments, which were already in the database. And what we then did is we fragmented this and looked for the fragment that we got from this hexose and compared it to all our standards. And I'm not going through all the data, just to say that basically we were able, with these massive amount of data, but actually these can be accumulated in really very short sequence of time, we could identify that these 3 peaks here obviously have all the same core structure, as I said before. We knew this already, and we could confirm that as well. So that was exactly what they suggested us do. But that the 3rd hexose here was a glucose as well, and that in this case here it's linked to the 3 position, here to the 4 position, and to the 6 position. We could tell exactly which one was which. And so we think actually for this sort of application in plant glycosides, which will always be very, very heterogeneous, this new sequencing strategy, including ion mobility, actually might be quite useful. And at the moment, actually, I would say we're extending it to polysaccharides as well, because we think so there are a lot of areas where carbohydrates are very important, but you don't really have the biosynthetic knowledge, you don't have really the standards you have for the N and O glycans, and then this sequencing might be quite useful. And it points towards a very universal sequencing strategy, so that maybe ultimately we just use this and we just throw all the standards out. I mean, we're not there yet, so I wouldn't do that, but that is a possibility. Just one more, under other applications, this is another paper we actually have colleagues of ours. It also works, if anyone is interested in heparan sulfates, we had colleagues who had heparan sulfates, which looked all very similar, but different in glucuronic acid or iduronic acid. So you can see here, these are the uronic acids. And you can get a lot of diversity by having, actually, these are all uronic acids, these are all glucuronic acids. This group had all the permutations, and we could actually distinguish them again with ion mobility. So again, somewhere biosynthesis doesn't really help you very much, standards don't really help you very much, but with ion mobility we can actually identify these and distinguish between them. And then one last, and this is from David Harvey at Oxford, who's actually an expert at ion mobility. I should say I haven't maybe given enough credit to colleagues who've really worked on this for a long time. David Harvey's really one of the people who was one of prominent. What he shows in this article here, again we've referenced it in our BBA. I was a bit too lazy to get his reference out. But basically ion mobility, where you can actually separate ions by mobility, but also M over Z. What you get is these what they call bins. So this sort of heat map is meant to show where the ions are. So these are the peaks. The colors are the peaks, and they're plotted against the mobility. This is ion mobility and M over Z. And because you've got 2 parameters over this, you get increased separation. You can then get mass spectra of all these different peaks. It's a sort of 2-dimensional technique. And what he can show is that he can really pull out minor components, very importantly often also from polymeric materials. You might have polymeric material here, but your minor component might be up here. you get very nice separation. So he, for example, says if you get send samples and it's got a lot of polymer from some column or something, ion mobility is a very nice way of actually pulling out, you know, sort of the active material. So what he's got here is a mass spectra of all these different bins, you know, from this one sample, and you can see they're all very different. He gets the doubly charged, this is all on N-glycans again, doubly charged, triply charged N-glycans, doubly charged glycopeptides, triply charged glycopeptides. You can pull out all these different species much better than with any other technique, because you've got this sort of capability of 2 dimensions. So it's a very nice way of doing ion mobility. So hopefully what I've shown you sort of a little bit sort of the state of the art in carbohydrate sequencing. And I know actually some of the instrumentation is obviously here. I know you use this sort of Rudden technology. You've just got a moldy TOF, so you can apply all these techniques. But also a little bit the future maybe with this ion mobility. And what I'm hoping is actually ultimately we have sort of de novo carbohydrate sequencing. The dream is really that in 10 years' time, someone can give us a sample, they don't have to tell us where it comes from, you know, and we just basically just put it into mass spectrometer, fragmented, do ion mobility and structural assignments through comparison with standards, comparison of CCS maybe with modeling data, which allows us to do some predictions, then assign all these fragments, and then use some computation methods to put it all together. Sort of a shotgun sequencing, universal sequencing approach that maybe works then for all glycans. So that is sort of the dream of the future. Yeah, but I think it is actually in principle now feasible by incorporating techniques such as ion mobility. And I think there are other techniques out there. At the moment we're collaborating with people who do IR, fingerprinting, so that's another method that you can integrate. So integrating all of this with these mass spec methods in the gas phase, the same time scale, I think will be very useful. So I'll finish there. So quite a long lecture today, and I'll thank you for your attention.